If you're doing all
following questions. Define, explain, and describe what your topic is. What's the issue you are discussing? What causes the issue you are describing? How and where does it occur? Is it only a result of human activity, or can it occur naturally? And are there any local examples? What is the ecological, well, I guess that should be what are the ecological impacts of the issue you are discussing? Why is this important, or why should we care? What are the actions that need to be taken to resolve or solve this issue? Short-term and long-term, what can individuals do? So these are presentations in the sense that you're going to do it as a slide deck, okay? So, but you're not actually presenting it. You're not recording yourself, anything like that. You're gonna share or submit the slide deck to me. They should be between 3 to 5 slides in length, okay? Probably 5 is optimal if you include a title page, okay? Presentation length should be timed for about 5 minutes in total, right? So if you keep that in mind, it should take you roughly 5 minutes. They've probably got the right timing for it. You'll need to reference your information and images using any style you like. You need a minimum of 3 different sources of information. Wikipedia can give you leads, but Wikipedia cannot be one of your sources. Okay? You're going to hand in the following: a soft copy of your presentation, Google Slides if possible, but other formats are okay. If you want to use Prezi, that's fine. If you want to use PowerPoint, that's fine, as long as I can open them in a Windows 10 PC. So if you're going to do it in, what's the Mac program called? Keynote or Pages, you're going to have to convert it into a format that my Windows 10 PC can read, because I can't open Pages files and I can't open Keynote files, okay? A hard copy of your references in any of these 3 formats, okay? There are 25 of you in class. There are 30 different topics, right? So everyone should be able to pick a topic on their own without overlap, okay, is what I'm hoping for. So I'll set up a Google Doc that I'll also add to this, where you can write down your name and the topic. Okay, so let's create a doc here. So it's going to be ecosystem disaster topics. Okay, and it's going to basically say name. Oops, not there. Topic. Then 1. And you can just list your name and your topic here. So the doc looks like this, right? So if you could pick any disease. So diseases in general that hit ecosystems, right? I don't know that we have, yeah, number 21. So it could be a disease. I would prefer that the disease be something that doesn't impact humans, but it impacts ecosystems. So for example, one of the diseases was a fungus that completely devastated the banana plantations. Well, if you want to pick a different one, you can, but you have to check with me. But if you're using one of these, then I only want 1 person per topic, okay? I realize 5 and 6 are kind of related. But I think there's enough differentiation between the two that we can basically talk about the two topics separately. It's on Classroom, right? I haven't finished creating it yet. I have two copies. Copy. Okay. So share. Yeah, it works. So if you just go to where it says untitled documents, right? So if I share the tab, when you open it, you're going to see this untitled document. I don't know why the title didn't transfer. But if you click on it, it should open up to this. Test for what? Genetics? Same length? Okay. I haven't figured out the value, what I'm going to mark it out of yet, which is why it says ungraded. Due date. When are you guys thinking? So let's look at a calendar. So what date would work? Looking at this, today's the 8th. Tomorrow is BFF day, remember. Grant, you've got your retest. And Nabil, oh no, he's in the other class, never mind. The date for what we're talking about is the date for the presentation. And today we're going to do the genetics test review. That's what we're doing today. So when do you want this to be due? Looking at that calendar. Because I haven't assigned the thing yet. So Ecosystems Disasters Project, the form should be right here. It's not a form, it's a Google Doc. So it's right here now. I haven't posted it until now. Okay, back to the calendar. So the 16th is one suggestion. Which will make it next Wednesday. Yeah. Does that work? Okay, so let me just edit that. I'll add the due date. You're on the 16th. Remember that the course ends on the 18th. Have one more what? No, probably not. One more assignment after this? No. The last unit, I'm probably not going to have any time to give you an assignment. So it'll what it'll be based on is my perception of your participation. Yeah, not even. Because if this is coming in on the 16th, I'll have 2 days to mark 60 papers. Oh, okay, hang on. Share. Sorry, it's taking a while to load the. Okay. Get link. Should be editors. Done. Kahoot for Prime? No. I could do Kahoot for knowledge, but not Kahoot for marks. I'm not quite willing to go down that route. So the last unit may be something where basically I just don't evaluate at all. It's more of a observation than anything else. I may run a Kahoot just to make sure that you guys understand the material for review, but I'm not going to do it as a marks, no. Okay. So that's that. Okay. The other thing I wanted to talk about, just so that you've got it, I've put up, this is not an assignment. This is just for you guys to practice on. Yeah, sure, Kirsten. Okay. All right. If you look at population dynamic problems, okay, which was posted this morning, okay, so these are problem questions for you to practice on for both exponential and logistic growth problems. Once it loads. So I'm at just questions I copied out basically out of the textbook. And also some density and population change questions for you guys to try to make sure that you understand the math. Okay, so it's not mark recapture, etc., stuff like that. This isn't for marks. This is just for practice. Okay, just to be sure that you understand how to do the math questions in this unit. Because although this doesn't count for marks for me, next year, if you're taking bio, there will be an ecology section where they basically expect you to know how to do this kind of stuff. So you should probably take a look at these questions. Okay, so it's not for marks, but it is there so that you guys can get an idea of whether you understand the mathematics behind these kinds of questions or not. Okay, so that's this. This is what we'll talk about on Thursday. Okay, and that Thursday we should be done the unit. Okay, ecosystem disasters project is there. I think that's it. Let's see, I can close that. Close that. You guys are right in there. Okay, so let's take a look at the genetics review stuff. So the genetics study guide. Okay. If you look over the review, is there anything that you have questions about? Anything that you want cleared up? Okay, one of the things I really want to be sure you guys can do is the following. So let me go back. Genetics. See if I can find the question. It's not there. It's not that. Nope. It's probably under my tests. Maybe that one. There's the question. My document. Oh, crap. Can I just paste it? No. Okay. How to do this? Do you guys have access to your textbooks? Okay, so what I need you guys to do, because I can't, what I've got here is a question I want you to try for a genetics review. Okay, so I'm giving you a chunk of DNA. Okay, from this chunk of DNA, write the mRNA sequence that will be transcribed from this fragment. Circle the starter and stopper in your mRNA sequence. Write the sequence of the amino acids that will be encoded in translation, and use the mRNA codon table. I can't drop the image into this Google Doc, so you're going to have to go to MyNelson and use the table there. So the table, if you go to MyNelson, it should be on page once it loads. Okay, if you go to section 7.3, okay, maybe it's not in 7.3. There, okay. On page 317, okay, figure 7 gives you the genetic code, okay? And table 3 gives you the full name if you wanted to use a full name. But what I want you to do, from this strand of DNA, write the mRNA that you're going to get. Remember that you need to start from the 5N of the messenger RNA when you make this, okay? And then try to figure out what the amino acid sequence would be using the mRNA codon, okay? Well, what I can do, Abby, let me see. Let me just go. We're going to call this practice problem. Protein synthesis. I'll share it with everyone. To anyone with a link. Copy the link. I'm going to drop the link into the chat. Yes, so it'll come off the DNA from the 5N, and then the ribosome will start at the 5' end and read it towards the 3. So you should be able to see the question there. Don't do C. I mean, that's pretty easy. Just do 2A and B. And what I will do then, Abby, is I will present my Nelson tab. And I can show, there's the codon table that you can use. Okay. Sorry, I can't share both screens at once. So give that a shot. You can do it on a piece of paper. You can do it in a different doc. That's fine. Just see if you can get A and B, and let's see how you do. Because there's going to be a question very similar to this on the test on Thursday. Abby, can you see the table from my share screen? Okay. So I'll give you, what, 10 minutes or so to try and figure this out? And remember, this is the DNA. And for this question, for these kinds of questions, you don't need to worry about introns and exons. Just assume this entire sequence is going to be transcribed. Pretend it's a bacteria. Okay, so the genes are continuous. I'm going to mute myself so you can't hear me breathing. Remember for 2A to label your 3' and your 5' in the mRNA. When you finish part A, just raise your hand, and then I'll start typing the solution when the majority of you have finished. Okay, 4, 5, So it looks like about 10 of you are working on this, because 5 of you are looking at the question. We're up to 11, 12. Okay, it's better. 3, 4, 5, 6, 7, 8, 9. Okay, so the first thing I'm going to do when I'm solving this, right? So share this tab. I'm going to start by labeling the ends. So since this is a 3 end, this has to be 5 end. Okay. Then we just follow along. The G has to go to a C. Oops. The A has to go to a U. The T has to go to an A. The A has to go to a U. The C goes to a G. Second C goes to another G. Then I'll have 3 A's in a row. Then a C, another A, C, another A, U, a C, E goes to an A, C goes to a G, A goes to a U, T goes to an A, C goes to a G, T goes to A, T goes to A, 3 prime. Okay? You can lower your hands now. Okay? So that's what the mRNA sequence is going to be, right? You know it's mRNA, so you have to replace all the T's have to become U's. So anywhere that you would have normally put in an T, you have to put in a U instead, okay? Yeah, that's fine, okay? Now, the next thing it says is circle the starter. Well, if we look at the mRNA codon table, once it loads, right? The starter, right, most of the time I said was AUG, okay? So methionine. So that being the case, if you look at this sequence, what you see right here, where's my highlighter? There it is. There's my starter. Okay, that's my starter. Okay, which means that this sequence is going to break up into codons from here. Which means there are 2 bases ahead of the start, which aren't going to be used. And there are 2 bases at the end, which are not going to be used. Now, the stopper has to be either a UAA, a UAG, or a UGA. Well, there's a UAG right here. So if I highlight that, okay, so there's my start and there's my stop. Okay. Now all I need to do is write the sequence of amino acids. So all I'm going to do is I'm going to use the codon table here, right, and figure out what each of these codes represents the codon table, and I could write right underneath it the names of the amino acids. So if I do that, we know that AUG is methionine. Oops, then GAA, so GAA is glue. Which is glutamic acid. ACA is threonine. CAU is histidine. CAG is GLN, which is glutamine. And then UAG, which is a stop.
Yeah.
Okay. So your amino acid sequence should be methionine, met-glu-thi-his-glin stop. Okay. And the key point with this one it worked out because I had 3 here, which meant this was 5, so I could read the protein sequence out in this direction. What would I have had to do if this was the other way around? If, as an example, instead that was 3, and that was 5, how would the mRNA sequence then change? Yeah, you have to go 5 and read it from this side. It will go AAG, AU, blah, 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 blah. And you'll be reading it this way. Okay? So when you're doing these questions, you have to check which end is 3 and which end is 5. Otherwise, because if you ignore the directionality, the anti-parallelness of DNA, you're going to run into a problem because you may read it the wrong way. Okay? So I want to be absolutely clear. Everyone understands that about this kind of question. So be aware, you know what end is 3 and what end is 5 so that you can write the mRNA in the right correct order. Also notice, you do not need to, unless the question asks you, you do not need to give me the tRNA anticodons because the RNA table, right, this table automatically goes from the mRNA to the amino acid. So you do not need to provide the transfer RNAs. Unless the question says, also include the transfer RNA anticodons in your answer. Okay? So be aware of that. If I just give you the mRNA and ask for the amino acids, don't give me the transfer RNA. I'd have to deduct points in that case. Okay? And then C, the question kind of depends if we're talking about a eukaryote or a prokaryote. In a prokaryote, where do transcription and translation occur? Remember that a prokaryote has no nucleus. Yes, but where? Yeah, cytoplasm. Okay, but in a eukaryote, where do transcription and translation occur? Nucleus and cytosol. Yeah, okay. So be aware if we're talking about eukaryotes or prokaryotes, right? If it's not clear, during the test, ask. Because I can't remember if I made that differentiation or not. Okay, let's see. Do I have any other practice questions for you guys? That's okay. Test 3. Those are okay. What are advantages that prokaryote cells have for DNA replication over eukaryotic cells? When we're talking about protein synthesis. Yeah, okay, that's the big one. They have no introns. Okay, that's one advantage. What advantage does the fact that they have circular DNA also give them? What else do they lack? No endpoints, which means that they don't have what occurring? What do we call it when a eukaryote loses the ends, the 5' end of a chromosome? So you need telomeres to fix it. Remember the term chromosomal erosion? Okay. So that's something that cannot happen in a prokaryote because they don't have ends. Okay. Let's see what else I can ask. In DNA replication, what's the direction of elongation? Is it 5 to 3 or 3 to 5? Do you remember? DNA polymerase adds new nucleotides to which end? Yeah, it's 5-3, always. Yeah, exactly. How does a repressible operon differ from an inducible operon? Okay. And an inducer? Okay, that works. Let's see. No, not that one. Okay, let's go through. Where is it? The genetics review. Okay. So these are the main guys I want you to know in terms of their contribution to discovering that DNA is the main, is the molecule of genetic inheritance. Okay. So what did Hump Morgan figure out? So that was Thomas Hunt Morgan, the guy who worked on fruit flies with their white eyes and red eyes. Okay. You should be able to tell me how Griffith's experiment works and what the significance of his experiment was. You should be able to tell me what was significant about what Avery, McCarty, and McLeod discovered, how Hershey and Chase's experiment confirmed Avery, McCarty, and McLeod, Chargaff's rules, influence of Pauling, Franklin on Watson and Crick, be able to describe the Michelson and Stahl experiment, as well as Beadle and Tatum. Okay, so Griffiths, Michelson, Stahl, Beadle, Tatum, Hershey, Chase. Those guys, you need to be able to describe their experiments. Elwin. Or did you raise your hand by accident? It's not here right now. Okay. All right. You should be able to describe the modern model of DNA. Okay. Understand how DNA replication works. And definitely, you should know the names of all the proteins and enzymes that are involved in DNA replication. Elwin, I saw your hand go up for a second. Did you have a question? Okay. Okay, you should be able to explain why you have leading and lagging strands, why synthesis only goes in one direction, and how Okazaki fragments play a role in the lagging strand to make a new DNA strand. Okay. You know what a telomere is, why they're needed. Okay. You understand how errors or mutations are corrected, and which enzymes are involved in that correction. You understand what the one gene, one enzyme, or one gene, one polypeptide hypothesis is. So this one, DNA, we just did, right? We did an example of that on the practice protein synthesis question. So you know how to read the DNA mRNA code and using a codon table, translate the sequence into amino acid. Okay, and this is key. Set your mRNA the correct direction, 5 to 3, so that you're translating correctly. Okay, you know what the codon for the start codon is and how to find the 3 stop codons. Okay, you understand what's involved in transcription, so how we go, so you're able to tell me what the steps are and the names of the enzymes. Okay. You understand what a promoter is, so the purpose of a Tata box, and determine how much DNA strand is the coding strand, and how to determine which strand is coding and which strand is template. You understand things like post-transcriptional processing, so the 5' cap, the poly-A tail on the 3-ended intron removal. You can understand why alternative splicing could occur. You understand the role of tRNA in translation. You understand how translation works inside the ribosome. You can understand, I probably won't have you guys do this actually, because I'm not going to have a diagram in the, I'm not going to have a diagram in the test that you have to label. So I wouldn't worry about labeling diagrams. But you should be able to summarize what's going on. You should know what slide 36 implies. I know what the 2 types of single gene mutation are, point and frame shift, and how they affect the gene. I understand some causes of mutations, and you understand how inducible and repressible operons work. LAC and TRP are specifically the 2 that we want to talk about. Okay? All right, it's 9:58, so it's 10 o'clock. Take a 10-minute break. We'll get back afterwards, and I'll give you time then to study on your own. Do you want questions from the textbook, or do you just want to go over your own notes and such? Okay, looks like notes will have it. Okay. So when we come back in 10 minutes, you can review your notes. All right. So I'll leave you be for the next half hour, I guess. No, longer than that. 40 minutes. So I'll be muted. I think I'll be marking GitHubs. All right. If you need anything, let me know. Is there a rescue anywhere near you? An animal sanctuary of any kind? Karema, the unit review starts on page 414. Just ignore the questions on anything on genetic technologies because we never covered it. But it starts on 414. So if it talks about electrophoresis or DNA sequencing or R-flips or PCR or restriction enzymes, things like that, we didn't touch any of that stuff, so don't worry about it. All right, guys, it's 51. Pity.